Close your mouth right now and take one slow breath in through your nose. Feel the air move along the floor of your nasal passages, cool against the tissue, pulling slightly at the back of your throat. You think you just delivered oxygen to your lungs. You did something else entirely. In the time it took that single breath to travel from your nostrils to your chest, it reached up into your skull and changed the electrical rhythm of your brain. Not metaphorically, measurably. The act of pulling air through your nose, rather than your mouth, just synchronized the firing of neurons in regions that have nothing to do with smell, nothing to do with breathing, and everything to do with memory and fear. And the strangest part is that it only works in one direction. Breathe in, and the effect appears. Breathe out, and it reverses. Switch to your mouth, and it vanishes completely. For most of the last century, breathing was treated as plumbing. Air goes in, oxygen crosses the alveolar membrane, carbon dioxide leaves, the diaphragm resets. The brain was considered a passive customer of this exchange, taking the oxygen it needed and otherwise ignoring the mechanics. The idea that the path the air takes, nose versus mouth, could reach into the cortex and reorganize neural timing would have sounded like mysticism. It is not mysticism. It is the consequence of an accident of anatomy that connects the air in your nose directly to one of the oldest structures in your brain. Here is the first mechanism, and it is mechanical in the most literal sense. When you inhale through your nose, the air does not flow silently past the tissue. It deflects, it tumbles, it presses against the lining of the nasal cavity. Embedded in that lining are the dendrites of olfactory sensory neurons. And those neurons are not only chemical detectors waiting for odor molecules. They are also mechanical detectors. They respond to the physical force of moving air. The pressure of the airflow itself bends them, and bending them makes them fire. This was shown directly. In 2011, a team led by Daniel Wesson, then at Case Western, demonstrated that olfactory sensory neurons in mice generate electrical activity in response to the mechanical pressure of airflow, independent of any smell. The nose is not just a chemical sensor. It is a pressure sensor wired straight into the brain. The detail that makes this work is the geometry of the molecule doing the sensing. The olfactory receptors involved are not exotic. They belong to the same broad family of receptors that detect touch and stretch throughout the body, and they sit in a membrane that deforms when air pushes against it. A puff of air during inhalation flexes the cell, opens ion channels, and lets a current flow. There is no odor required, no chemical key fitting a lock. The trigger is force. This is why a completely odorless breath of clean air still produces a burst of firing in the bulb. The neuron is reporting the arrival of the breath itself, the way a windsock reports a gust. And because that firing is tied to the moment of inhalation, it is automatically rhythmic. The rhythm is not generated by the brain and imposed on the breath. It is generated by the breath and imported into the brain. And the structure those neurons feed into is the olfactory bulb. This is the part that matters, because the olfactory bulb is not a remote outpost. It sits at the base of the frontal lobe. And it is one of only two places where the outside world makes nearly direct contact with the brain's interior. Every time you inhale through your nose, you send a rhythmic mechanical pulse into the olfactory bulb. Every time you exhale, the pulse stops. The bulb, in other words, receives a clean, repeating signal that tracks your breathing precisely. Breathe at 12 breaths a minute, and the bulb pulses 12 times a minute. The brain now has a clock, and the clock is your breath. A clock by itself does nothing. The question is whether anything downstream listens to it. And this is where the story changes, because the answer came from inside a human skull. In 2016, Christina Zelano and Gottfried, working at Northwestern University, published a study in the Journal of Neuroscience that did something rarely possible. They recorded electrical activity directly from inside the brains of patients who had electrodes implanted for epilepsy surgery. These were not scalp readings filtered through bone. These were electrodes resting on the hippocampus and the amygdala, the structures responsible for memory and emotion. And what they found was that the natural rhythm of breathing was stamped onto the activity of both. The oscillations in the amygdala and the hippocampus rose and fell in lockstep with the breath. The brain's deep emotional and memory centers were keeping time with the air in the nose. Then they did the critical test, the one that separates a coincidence from a mechanism. They had the patients breathe through their mouths instead. The entrainment collapsed. The rhythmic coupling between breath and brain weakened dramatically the moment the air stopped passing through the nasal passages. The signal was not coming from the rise and fall of the chest or the movement of the diaphragm. It was coming specifically from airflow across the nasal tissue. No nose, no signal. The mouth breathes just as much air, moves the same diaphragm, delivers the same oxygen, and the brain's rhythm does not care. It only responds to air in the nose. Now think about what that means for memory. The hippocampus does not encode memories at a constant rate. It works in cycles, and there are windows within each cycle when it is more receptive to laying down new information, and windows when it is busy replaying what it already holds. If the breath is setting the timing of those cycles, then the phase of your breath, whether you happen to be inhaling or exhaling, should determine how well your brain captures a given moment. Zelano's team tested exactly this. They showed subjects images of fearful faces and asked them to identify the emotion. When a fearful face appeared during a nasal inhale, subjects recognized the fear faster than when the same face appeared during exhale. The difference was real and repeatable, and memory followed the same pattern. Objects shown during nasal inhalation were remembered better than objects shown during exhalation. The moment of the breath was acting as a gate on what the brain absorbed. Sit with the strangeness of that. Your ability to read fear on another person's face, and your ability to remember what you are seeing, fluctuate several times a minute according to whether you are breathing in or out through your nose. We imagine perception as continuous, a steady stream. It is not. It pulses, and the pulse is your breath. There is a way to feel the edge of this yourself, and it costs nothing. The next time you are startled, notice what your body does first. It does not exhale. It gasps, a sharp inhale through the nose. For a long time, that was explained as simply grabbing oxygen for a fight or flight response. But the timing is wrong for that. The oxygen is not needed in the first quarter second. What the sharp nasal inhale does immediately is flood the amygdala with that mechanical pulse at the exact instant the brain most needs to process a possible threat. And Zelano's data shows the amygdala is most responsive to fearful stimuli precisely during nasal inhalation. The gasp is not fetching air. It is opening the gate on the fear circuit at the moment of danger. Your body has been using the breath as a cognitive trigger your whole life, in the one situation where milliseconds matter most. But there is a second mechanism operating at the same time, and it works through a different rhythm entirely. The olfactory bulb does not just pulse with the breath. It generates fast oscillations in the gamma range, roughly 30 to 100 cycles per second. And these fast oscillations are nested inside the slow rhythm of breathing. The slow breath rhythm acts as a carrier wave, and the fast gamma activity rides on top of it, strongest at particular phases of the breath. This nesting of a fast rhythm inside a slow one is called cross-frequency coupling, and it is one of the fundamental ways the brain organizes information across regions. The slow wave says when, and the fast wave says what. Nasal breathing provides the slow wave that organizes the fast computation. Mouth breathing does not, because the mechanical drive on the olfactory bulb is gone. This is not confined to the nose and its immediate neighbors. The olfactory bulb projects to the entorhinal cortex, and the entorhinal cortex is the main gateway into the hippocampus. So the rhythmic signal that begins as air pressure in your nasal cavity propagates along a real anatomical chain, from olfactory neurons to the bulb, to the entorhinal cortex, into the hippocampus, and out to the amygdala. Each link is a known pathway. The breath is not influencing the brain through some vague systemic effect. It is riding a specific cable that evolution laid down hundreds of millions of years ago, back when the dominant sense was smell, and the entire forebrain was built around processing odor in time with sniffing. Here is the part of this story I find poignant. For most of the 20th century, the olfactory system was treated as the embarrassing relative of the senses, a primitive holdover, the least interesting input to a human brain that had supposedly graduated to vision and language. Researchers who studied smell were studying what the brain had left behind, and yet it turned out that this discarded system was carrying a signal that organizes memory and emotion in structures we consider the height of human cognition. The same bulb that detects whether bread is burning is the one providing the rhythmic scaffolding to the hippocampus. The unglamorous sense was never demoted from the architecture. It was hiding the timing mechanism the whole time. And we only found it because someone thought to record from inside a human skull while the person simply breathed. That evolutionary history is the key to why this exists at all. In a mouse or a rat, sniffing and thinking are nearly the same activity. The animal samples the world in rapid rhythmic sniffs, 5 to 10 a second, and its brain processes each sniff as a discrete packet of information. The breath rhythm and the cognitive rhythm are fused. Humans inherited that architecture. We breathe far more slowly, and we are not navigating by smell, but the wiring that couples nasal airflow to deep brain rhythm was never removed. It is still there, still running, still stamping the timing of your breath onto your memory and your fear circuits. A piece of ancestral machinery operating quietly underneath the brain that thinks it has moved on from smell. And here is where the evidence has to be handled carefully, because the field sits at different levels of certainty. That nasal airflow entrains brain oscillations is solid. It has been recorded directly from human brain tissue and replicated. That the phase of the breath modulates memory and emotion recognition is well supported by Zelano's behavioral work and by subsequent studies. What remains less settled is how large these effects are in everyday life. Outside the controlled setting of a lab showing flashcards, the brain is not a slave to the breath. Attention, novelty, and a dozen other forces shape what you remember. The breath is one rhythm among several, an influence, not a dictator. Anyone who tells you that breathing through your nose will transform your memory is overstating a real but modest effect. The honest claim is narrower and stranger. The timing of your breath measurably biases the timing of your cognition, and that bias is gone the instant you switch to your mouth. There is also the matter of speed, and this is where slow breathing enters. The coupling between breath and brain works at whatever rate you breathe, but the rate changes the character of the signal. When you breathe slowly through your nose, around 6 breaths a minute. The slow carrier wave stretches out, and the gamma activity nested inside it has longer, cleaner windows to organize within. When you breathe fast and shallow through the mouth during stress or anxiety, that organizing rhythm is both absent because the air bypasses the nose and disrupted because the timing is erratic. This may be part of why slow nasal breathing has such a reliable calming effect that it appears in nearly every contemplative tradition independently, from yogic pranayama to monastic chant. Those traditions did not know about olfactory bulb entrainment. They discovered empirically that a slow breath drawn through the nose settles the mind, and they built entire practices around a mechanism it would take neuroscience thousands of years to begin to image. The instruction was right long before the explanation existed. There is a further layer, and it concerns nitric oxide, though not in the way it is usually described. The paranasal sinuses continuously release nitric oxide gas into the nasal airway. When you breathe in through your nose, you carry that nitric oxide down into your lungs, where it relaxes the blood vessels around the alveoli and improves the efficiency of oxygen uptake. When you breathe through your mouth, you bypass the sinuses and you lose that nitric oxide entirely. So nasal breathing is doing two separate things at once that mouth breathing cannot. It is mechanically entraining your brain through airflow, and it is chemically improving your blood oxygenation through a gas your own skull manufactures. These are independent mechanisms riding the same breath. One is electrical and one is molecular, and the mouth defeats both. Now consider the chronic case, because a single breath is one thing and a lifetime of breathing is another. Many people breathe through their mouths most of the time, especially while sleeping, often without knowing it. If the nasal rote is what couples breathing to the rhythms of the hippocampus and amygdala, then chronic mouth breathing means a brain that spends years without that rhythmic input during a substantial fraction of its hours. The direct long-term consequences for human memory have not been cleanly measured, and it would be dishonest to claim they have. But the mechanism gives us a reasonable hypothesis. The structures that the breath entrains are precisely the structures most involved in consolidating memory during sleep. And sleep is exactly when many mouth breathers are not using their nose. The physics supports the concern. The longitudinal human evidence is not yet in. That gap is where careful science lives, and it is more honest to name it than to paper over it. Pull the layers together and a single picture emerges. The breath does not affect the brain through one channel. It affects it through at least three, running simultaneously on the same inhale. There is the mechanical channel, where airflow bends the olfactory neurons and pulses the bulb. There is the rhythmic channel, where that pulse propagates through the entorhinal cortex into the hippocampus and amygdala and sets the phase of memory and fear processing. And there is the chemical channel, where sinus nitric oxide rides the same airstream into the lungs and tunes oxygen delivery. These are not the same mechanism described three ways. They are three distinct physical processes that happen to share a trigger, and the trigger is one quiet breath drawn through the nose. The mouth, for all that it moves the same volume of air, engages none of them. It is the difference between a wire that carries signal and a wire that has been cut, even though both are made of copper and both carry current. The path is everything. What is not in question is the immediacy of the effect. You do not have to train for weeks. The coupling appears within the first breath. The moment air moves across your nasal lining, the olfactory bulb pulses, the entorhinal cortex relays, the hippocampus and amygdala fall into step, and the gamma oscillations begin nesting inside the rhythm of your breath. 60 seconds of nasal breathing is roughly 12 to 16 of these cycles. 12 to 16 pulses of rhythmic organization delivered to your memory and emotion circuits. 60 seconds of mouth breathing is none. The difference is not in the oxygen, which is identical. The difference is entirely in the path. There is something almost unbearably elegant about where this leaves us. The nose was understood for most of medical history as a humidifier and a filter, a piece of preconditioning equipment for the lungs. It is that, but it is also a transducer, a device that converts the physical motion of air into an electrical rhythm that the deepest parts of your brain use to organize themselves in time. Every quiet nasal breath you take is a metronome tick delivered to your hippocampus. You have been keeping time with your own breath your entire life, and you never heard the beat, because the beat is not a sound. It is a pattern of firing, written into the same tissue that holds your memories and reads the fear on the faces around you. So go back to the breath you took at the start. The slow one through your nose, the one you thought was just oxygen. In the second or two it lasted. It pressed on the mechanical sensors in your nasal lining, fired the olfactory bulb, sent a pulse down a pathway 400 million years old, and briefly synchronized the firing of the structures that decide what you will remember of this moment and how quickly you would recognize a threat. It did all of that silently, automatically, without your permission and without your awareness. Close your mouth and do it again. Not because it is a wellness ritual, because it is the only way the air reaches the part of you that keeps the time.